I'm going to start part 2 of my talk. This time I'll be focusing a little bit more on the larger picture of GHGA, the workflows in it, and how those workflows should be organized. So like Julia mentioned in the introduction, GHGA is the German Human Genome-Pheome Archive. Essentially, it's a lot of letters to describe a coalition of universities, hospitals, and computation centers dedicated to the storage of OMICs data, which is genomics, transcriptomics, and other types of relating to the genome data, facilitating research across Germany. So luckily, it's not just me working on this. There's a large number of consortia and institutions and experts and a great board of directors guiding this project to standardize the way that genetic health data in Germany is handled. So GHGA is trying to take this national expertise to establish an infrastructure for human omics data, facilitating the improvement of health and medical research in Germany. So the goal is to develop a platform for long-term fair data archiving of human data, connecting major centers in Germany, allowing them to share and process data in a safe and secure way. We hope to establish and offer standardized data processing workflows, build an ethical and legal data use framework for data sharing, protection, using controlled access management and community-centered data sharing platforms, and to establish strong interfaces within the larger international community outside of Germany as well. So what does this actually mean, and how can GHGA impact someone's daily life? So GHGA is hoping to bridge the gap between research and clinical care. The goal is to provide technical and legal infrastructure needed to confidently share data between patient groups, clinics, hospital, diagnostic labs, and facilitate research such that regardless of your hospital setting, that you can benefit from the best research and science that science has to offer. So GHGA can offer a place to store your data and allow researchers to apply and collect relevant data and potentially reanalyze samples as the scientific base knowledge improves. GHGA is hoping to improve the feedback loop of patients and healthcare to research, improving tools. Those tools improve patient care, that patient care improves the research, and so on and so forth. So why do we actually care, and what good is it to collect and sequence data? Why go through this effort at all? And the reason is that a collection of health data allows for improved quality of care and improves research capability. So we'll focus on a couple of quick examples. So first, we can imagine, for the case of rare disease, 4 patients scattered across Germany, maybe individuals in Cologne, Leipzig, Munich, and Kiel, all of which have a rare disease and an unknown cause. It's undiagnosed, and each hospital and set of doctors look at all of their records and can never find something that quite fits the story. But if you can share this data, you can narrow it down and in fact align the differences between these individuals such that you can clearly see a commonality across these patients. So even though they all have 4 or 5 rare, potentially causing mutations, all 4 of these patients have the same type of disease, they all have the same health outcomes, and they all have the same variant at this particular location. So by combining the data that we have in a safe way allows us to lead to better diagnoses and better understanding of disease. There are other benefits with respect to cancer. How do tumors differ across patients and types? To common disease, are there parts of the genome that protect against heart disease? All else being equal, how do different genes add or detract from susceptibility to things like obesity or Alzheimer's or COVID? And then lastly, what about future NIC? If we want to worry about our future healthcare, we want to benefit from research that hasn't been done yet. So if in the next year a new breakthrough or tool is developed, we want to be able to retroactively apply this to patients that were unable to originally benefit from these discoveries. So hopefully that kind of outlines some of the utility of collecting sequencing data and using it for research and diagnoses. However, I want to focus on some of the potentially more important aspects, like how do we keep it and why is this happening? So everything we talked about is only possible within the realm of a legal framework. And I'm not a lawyer, so I will try to choose my words very carefully, but GHGA luckily has a vast team of people who are much smarter on this aspect than I am. But there are laws requiring the planned management of how to handle genetic data. So the way storing data this sensitive has to be done legally and correctly. Genetic health data is extremely sensitive, not only to the individual, but to their families. So whether, I like it or not, I share genetic data with my brother, or genetic information with my brother. And so not only is my data my concern, but my data is his concern and vice versa. So at GHGA, we are really intently focused on precise reading and adherence to data protection requirements and focused on adherence to GDPR requirements. So GDPR compliance is a focus in every aspect of GHGA and how we regard sensitive data and how it can or cannot be shared. So we are focusing on strict definitions of data submitters, who controls the data, those who are requesting access to the data, and making sure that we have a clear-cut role for each of that. We also want to make sure that any consents relating to the patient are honored, because we want this to be bulletproof and clear from beginning to end. So getting away from the legalese and legal aspect of this, I've talked about FAIR data, and this is an acronym, and FAIR workflows, which is an acronym meaning findable, accessible, interoperable, and reusable. So it's essentially a set of international guidelines to make sure that data and workflows are serviceable for the research community. So we'll get into each of those in more particular pieces. So when something is findable, it's relative. So imagine Googling GHGA. I think there are around 85,000 hits, but if it's not on page 1, it's not findable. No one wants to go on page 10 of Google, and no one wants to go to page, to item 2 of Google. So we want to allow researchers to deposit and find data relative to their use case. It should be clear, obvious, and simple to find data relevant to your research question using clear labeling and other data labels. We want this data to be accessible. So it needs to be accessible through a well-defined protocol to allow for secure and transparent access. Because we are working with sensitive health data, the accessibility access is key. We have to have protocols in place to make sure that data access is only possible through data protection committees, data protection officers, to allow those that have a need for the data to have access for it. We don't want anyone, to have access to your genetic health data. There needs to be clear and important reason for that data to be shared at all. Thirdly, data should be, and workflows should be interoperable. So like riding a bike, it doesn't matter if you pick up a bike from the central train station or the museum, you want it to work and you want it to get you from point A to point B. So at GHGA, we aren't building a Tour de France style bicycle, we're building a city bike, focusing on something that is easy to use, something that is shareable, such that it leads to more consistent and standardized usage that still gets you to where you need to go. So we're working on building workflows that are easy to use across multiple locations, that allow you to use different computational setups, and the data processing reflects that ease. And lastly, that data should be reusable. So not only are big projects and big, monolith black boxes of workflows used, but we want the small internal parts to be reused and applied to different projects. So it's important to keep records and version control to make sure that you have an understanding of how your data is being processed, so that it is transparent at every step of the way. So I realize that workflows might not be a term that everyone uses in their everyday life. And so I want to define it as essentially a roadmap that is modular, made up of small pieces that fit together, so that if you know what goes into each step, you can know what comes out of each step. You can combine and reuse these pieces to build larger projects. So I apologize in advance to any pizza chefs in the audience, but as an example, we can think of making a pizza as a series of steps. So whether you are mixing cheese or kneading dough or cooking sauce, you can think of these as inputs to a step and outputs, some of which can be done in parallel. You can, for example, cook sauce and mix cheese at the same time, but some things can only be done after a previous step is finished. So unless you want a big chunk of raw onion in your sauce, you have to make sure that you chop your vegetables first. If you want to make sure that you have a pizza, you need to make sure that you work the dough before adding the cheese. So some of these steps are order specific and input specific, but some things can be reused as well. So for example, the mix in the bowl step is the same whether or not you're talking about dry ingredients, exclusive the water, or cheese. You know, you're just putting stuff in a bowl and mixing it up. However, things like cooking your tomato sauce is drastically different than baking, and they have different inputs and different outputs. So there's more than one way to make a pizza, and there's more than one way to process genetic data. So at GHGA, we aren't out to create our own workflow, but instead our job is to make processing data easy and clear. So the goal is to work, our goal is to work with existing workflow tools and workflow development systems to gather collections of tools that can be easily walked through to process to do different parts of the genome processing workflow. So from the first part of the lecture, we talked about read alignment being a step that happens before variant calling, but there are other steps such as quality control and trimming, or larger novel importance of how DNA's larger structures are different, as well as how do we report these differences and how do we annotate these changes. So at GHGA, our goal is to produce standardized configuration files, essentially give someone an input of how do we want a process to be run, and then let the individual processor use their particular computing environment to set up. So we would say we want to use reference A and tool B with dials set to X and Y, and it's up to the individual processor to use that basic setting and trust that that basic setting will give them the same results whether or not they're in Munich or in Berlin or Tübingen or Kiel or wherever else you might be. So back to the pizza analogy, we might say we want to use 100 grams of olive oil, but we don't necessarily care that you bought your olive oil at Aldi or Rewe, or if it's a 50-year artisanal blend. You just need 100 grams and we'll go from there. And it's not a very good analogy. But essentially, our goal is to collaborate within the larger bioinformatics community. So we don't want to generate a new workflow with new standards and have to convince people to use us because we're better. The goal is to convince people that using our workflows is what everyone else is using, and that it allows you to compare your data easier and more effectively across different groups. So getting back to the rare disease example I showed before, if all of those variants are processed using their own different workflows, it's a lot harder to line them up, and it's a lot harder to find the shared variants that I highlighted before. So the additional value of this is building on top of existing communities and existing standards such that people across the bioinformatic landscape and the genetic health world are using the same tools that they already use anyway in an easy to use way. Again, this is focusing on the city bike, not the Tour de France bike. And all this relies on open source, transparent documentation and understanding of how this workflow actually performs and why we decided to use the tools and settings that we did. So with that, I'm going to summarize GHGA into establishing a legal and technical infrastructure for processing genetic health data, that it's useful to have databases and data from different sources to improve research and clinical care, that data and the associated workflows should be fair, findable, accessible, interoperable, and reusable, and that we aim to please. That this is a community project, and that the goal is to make GHGA standard across Germany and make the choices we use clear and obvious to anyone. So if you want to follow more in our newsletter, Julia had a QR link in the beginning. There's a registration site once you follow this link. But additionally, if you find yourself as an expert and you really want to identify what GHGA can do for you, you can fill out this survey that we have. So this is generally for those within the genetic processing landscape as well, or already, excuse me. And so what do you want from your standardized workflows? What raw data do you actually want to do? Do you want to use matchmaking services to connect you and your samples to other hospitals and theirs, and what it is you'd have? So if you find yourself really having strong opinions, please fill out the survey. It will take around 5 to 10 minutes. Join the newsletter, and thank you very much for your time. I think we're running out of time for questions, but I'd be happy to answer whatever you got.
So maybe Nick, a question from my side. You're saying you want to use standardized workflows. Why do you think standardized workflows are so important?
I think that while there's a lot of very specific tasks that require a certain set of very astute, kind of performance enhancing dial setting, how do you set one tool to accomplish a specific task? That there are a lot of things when it comes to genetic health that can be improved by having a standardized process. So going back to the rare disease example, is that if each of these 4 hypothetical patients are all processed with different tools or processed with different dials set to different settings, that it's way harder to align them. And I can assure you that anyone who works in the bioinformatic world knows that it's a mess and difficult to combine results using different reference genomes or different annotation sets, which is essentially guidelines to how you define a gene. And so that understanding the benefits that you get for having standardized processing that standardized workflows brings allows for a broader utility across different healthcare sites. So we're certainly not in favor of telling anyone that their workflow is wrong. We just want to make sure that we are serving the broader community and allow them to take these results and dive into them with their specific tools, potentially on their own.
There is another question in the chat. So from Jana Mida, are you planning to integrate GA4GH packets in the context of your initiative?
Yes, absolutely. GA4GH is the Global Alliance for Genomic Health, and it's another international organization that tries to help improve the standardization. So there are plenty of people at GHGA who are working with GA4GH in mind. So we have a metadata team that is describing how our uploads and how the tools that go into the sequencing runs are defined. Within the workflows itself, we are focused on meeting GA4GH standards of open source and containerized workflows to make sure that they are consistent and with the international community.
Thank you very much, everyone, for joining. It's lovely to see so many of you here. Again, if you would like to have more information around GHGA and maybe other events that we are doing, we're also running a very interesting podcast series. Please have a look at our website or sign up for our newsletter. Thank you, Nick, again. Have a lovely day, everyone.
Yeah, thank you very much. Enjoy your day.